welcome to Bloodwork. I'm thrilled to be here today at the European Hematology Association Congress in Milan, speaking to Dr Katherine Bridge. Katherine is a group leader from the University of York, and today she's going to be talking to me about myeloproliferative neoplasms. Thank you for joining me, Katherine.
Thanks for having me.
So, can you first of all tell us what is a myeloproliferative neoplasm?
So myeloproliferative neoplasm is quite a long umbrella term for a group of chronic myeloid cancers. And so what that means is that in blood cancer, we tend to categorise them as either the lymphoids, so more sort of going towards the white cells, or myeloid, which can go more towards the red cells, like red blood cells, for example. So there are three types of cancers within the umbrella term of MPN, and all of these are very chronic, indolent, long-lasting cancers. And why it's important for us to study them is that, well, firstly, they're not curable. So for people with MPNs, with that diagnosis, that would then be something that they are living with for the rest of their life. And there are treatments that can help to alleviate some of the symptoms. But in some cases, and for some patients, they will then transform into a much more aggressive or acute myeloid disorder like AML.
Sounds like there's a real unmet clinical need here then.
Yeah.
So I know that in your recent work you've been focusing specifically on a type of MPN with JAK mutations. Is this quite a common mutation for these patients?
Yeah, it is. So it's a mutation that happens in the JAK2 gene, and these range between 50 to 80% of patients with MPNs can have these, and for one group of MPNs, the incidence is about 80%. So it's really it's a really common mutation.
Sounds like it's really important to find out what this is doing then, and this is where, of course, your fantastic work that's just been published comes in. So can you tell me what JAK2 is doing in these cells?
Yeah, so what we found is that JAK2 is able, this mutation is able to mimic hypoxia and the way that the cell responds to hypoxia. So in a normal, healthy cell, we know from our GCSE biology that cells need two things to survive. One is glucose and the other one is oxygen. So when a normal cell is maybe getting not enough oxygen, it's able to switch on this really clever mechanism that helps for more oxygen to be delivered to the cell, and also for the oxygen that's in the cell to be conserved. And that will enable the cell to survive through a period of having not enough oxygen. So that's really important that cells can do that. What the JAK2 mutation does is to mimic that state of being hypoxic. And what that means is that it's able to access all of those signals to send out to help it to grow and to proliferate faster.
So it sounds like this is really rewiring the whole biology of the cells. Is that what you're seeing? Is that how they're turning into cancer cells?
Yeah, that's right. So my lab works on the DNA and how gene expression is controlled in these JAK cells. And what we see is that this hypoxia mimicry that the JAK mutated cells are able to do really changes the genes that the cells expressing. And those genes are associated with how severe the disease is, how fast it progresses, and really critically, we have identified a set of genes which correlates with transformation to the more acute myeloid leukemias.
Okay, so is this gene signature going to be prognostic for these patients? Can you predict who is going to actually progress?
Yeah, so that's something that we definitely want to explore with this set of genes. I think from the therapeutic perspective, what's really exciting for us is that we found the proteins that are interacting at the DNA, which are controlling gene expression, are actually very different from what we would see in the normal physiological, let's call it, hypoxic response. And so that gives us hope that we can target this hypoxia mimic by targeting these proteins that are different from a normal, healthy hypoxic response.
Oh, that's brilliant. So this is looking like a true targeted therapy to spare the healthy cells in these patients as well then.
Yes.
And are there drugs already available for this? Is this a targetable axis you found?
Yes, so some of the proteins that are present in the JAK mutant cells in these complexes have already had drugs developed for them. So some of these are involved in the DNA damage response, which is a response that cancer cells are also very good at exploiting to help them not die, essentially. And so there are already drugs that are being used in other types of cancer and in other types of myeloid cancers like AML, which actually could, from what we found, have the potential to hold promise also for MPNs, and potentially even more so for patients where we see this gene signature. It may help to prohibit or to stop the transformation to a more aggressive disease.
That's brilliant. That's really, really fantastic. And I think that gives a lot of hope for our patients suffering from these MPNs. So what's next on the horizon in this project? How's it going to move forward?
Yeah, we really hope so. And I think as a lab, our goal is always, how does this actually change how cancer is treated? And so the next step is to start to do the experiments where, we target these proteins, we knock them down, can we switch off this gene signature that we've identified? And doing that in the lab, and then with a goal to start to work more with clinicians to see, could this be a new therapeutic opportunity one day?
Fantastic. Well, it sounds brilliant, and I'm really hopeful, and I hope you are too, and we shall follow with interest and see where this leads.
Thanks very much.
Thank you for joining me today, Katherine, and sharing your work. I hope you've all found that interesting. Please remember to like, subscribe, and if you've got questions for Katherine, just drop them in the comments below and we'll get them sorted for you. Thank you.